Nazia, am I audible now? Okay, so as per my record, today we were supposed to do the last chapter of syllabus. In fact, so it was plant growth and development. Am I correct? Last time we have completed respiration part, right? So let's start with the that chapter. So plant growth and development. So before starting the chapter, I want to tell you in this chapter was the most important thing is that is the hormones, plant hormone, that is the phytohormones and their function, right? So that is the most important thing. Other than phytohormone, there are small topics which are very important, like what is the plasticity and all that. We'll discuss that also, and short day plant, long day plant, and all that, right? So let's start with that chapter. Plant growth and development. Plant growth and development. Yes. Yes. So let's start with the plant growth and development. And first, we have try to understand. We have to understand that what is actually. Uh, oh, I won't share that. Okay. So look, Mazia. Today we are going to start with the first chapter. That is the what is the growth. So can you tell me what is the growth? Any idea? What is the growth, Mazia? How can we define the growth? So look, how we can define the growth. The growth is that is the increase in size or weight, right? And if size or weight is increasing, in the nutshell, if mass increase in irreversible increase in mass, that is called growth, right? And one more thing, when we talk about the growth in the living, this is intrinsic, means it comes from inside of the organism. How? Look, suppose that my height is increasing, your height is increasing, your size is increasing. It means our bones are growing, right? So something internal which is growing, right? So this is called growth. So start writing down growth. Increase in increase in mass or size and weight, size and weight of a cell of a cell or organ is called or organ is called growth. Full stop. Growth is growth is always intrinsic. Growth is always intrinsic. That is comes from inside. That is comes from inside in living organism. In living organism. Full stop. Now come to the phases of growth. Right? How many phases of growth are there? So we have divided. Now here we are, as we are studying the plant physiology. So we are talking about. We'll talk about the plant only, right? So phases of growth. When we talk about the phases of growth, right? So we divide it in three phases: formative phase, phase of enlargement, and phase of differentiation, right? So there are three phases. So when we talk about the growth phases, so the growth is divided in three phases. So start writing now. Phases of growth, right? Growth of the plant. Growth of the plant can be defined. Growth of the plant can be defined in three phases. Put the first subheading, that is formative phase, right? That is formative phase. Put the first subheading, that is formative phase, right? So formative phase. So what is actually formative? First, I'll tell you. So formative phase means formation of new cell. So where new cell formation will take place. Wherever the meristematic tissues are available, their new cell will be formed. Phase of enlargement, increase in the size of cell, right? And then phase of differentiation means division of the labor of cell. So that is the three phases of growth, right? So that's how the growth takes place. So Growth takes place in three phases. First, new cells form, then the cells increases their size, enlarges, and then they get the particular job, particular function to start. Formative phase. That is formative phase, cell formation or cell division phase. Cell formation or cell division phase. Here, the formation of cell takes place in this phase. Next point. It always takes place in root and shoot apex. It always takes place in root and shoot apex, which have meristematic tissues. Next point, phase of enlargement. In phase of enlargement, cell enlargement takes place. In phase of enlargement, cell enlargement takes place. Now, how the cell enlargement takes place? Actually, small we have already studied that when newly cells are formed. So what happened? Newly formed cells, vacuoles are very small. When cells increase its growth, like increase its size, so all small vacuole they combine and they make a central vacuole, and they push the protoplasm and nucleus aside, and that's how they increase their size. So please note it down. In cell enlargement, the development of central vacuole takes place. And that's how development of central vacuole takes place, and that's how cell increases its volume. For example, in watermelon, during cell enlargement, it increases cell size. Up to 3,50,000 times. 3,50,000 times. So look, during cell enlargement in watermelon, one cell increases its size. Every cell increases its size 3,50,000 times. Right? That's how that much enlargement, cell enlargement takes place. Right? So, full stop. This is also known as phase of elongation. This is also known as phase of elongation. Next heading, phase of differentiation and maturation. Phase of differentiation and maturation. Start noting down. The enlarged cell develop into a special or particular type of cell. Particular type of cell by undergoing a structural and physiological differentiation. Is it clear? Clear to both of you? Phases of growth. Keep on telling me. So then I'll be able to understand that are you getting my point or not. Look, so this is the growth curve. So what is the growth curve? Growth curve show the this is a graphical representation of growth of a plant, right? So complete life history, how does the plant grow, right? So I have plotted a growth curve here. In growth curve, in lower axis we have shown the time here, and this is the time, and in upper axis we have shown the growth, right? And now the graph is like this one. So in initial phase is called lag phase. During the lag phase, a seed germinate, new part formation takes place, germination takes place, it grow slowly. Then the next phase is log phase. In log phase, the plant start going exponentially. So during this is the young time period of the plant when it will grow, it will grow speedily, and then the graph's slope will be very steep. And after some time, the this is the decelerating phase. In decelerating phase, after the reaching at maturity, so what will happen? The growth rate will decrease. This is the decelerating phase. And at particular growth time phase, at particular time, what will happen? The growth will become steady, right? It will become constant. So this is called stationary phase. So we have divided this growth curve in lag phase, log phase, deceleration phase, and stationary phase. Please note down this graph. Note down the growth curve. Note down the diagram. And let me know, message me when you note it down, both of you. Done, Mazia? Alvia, done? Plotted the graph? Guys, keep on messaging me. Like when you do it, uh, message me, right? Start noting now. In growth curve, within this heading, growth curve, start noting now. It is the definition of growth curve. It is the graphical representation of it is the graphical representation of total growth graphical representation of total growth against time. Let's stop. The period of time in which growth takes place, the period of time in which growth takes place has been called grand period has been called grand period of growth has been called grand period of growth. Full stop. The graph show S shape. The graph show S shape or sigmoid curve. The graph shows S-shaped or sigmoid curve, right? So this was the growth curve. Now put the next heading, that is the condition for growth, right? So look, it's very simple. What are the condition for growth? So any plant, if it have to grow, so plant require what? First, it need nutrient, water, oxygen, light, temperature, and gravity. Please note down the condition for growth. So there are following condition of growth. There are following conditions of growth. First, nutrient. Second, water. Third, oxygen. Fourth, light. Fifth, temperature. Sixth, gravity. So look, nutrient, it takes from where? Soil. And we have already studied the chapter in detail, that is plant nutrient. And the second one is water. Yes, it requires water for the photosynthesis. Oxygen, it requires oxygen for the respiration. Light, yes, it do require light for the photosynthesis. Temperature, at optimum temperature only enzymes and hormones work, and at lower temperature or higher temperature enzyme and hormones do not work. Next, gravity. So in gravity line, please note it down in the gravity point. Gravity determine the direction of root and shoot. Gravity determine the direction of root and shoot. Next one. So next one is, that is, what is the this one? What is the development? So what is the development? Look, so there are certain phases of development. So what happened in development? We'll study the differentiation, de-differentiation, and re-differentiation. So first, let's talk about the development. Development is nothing but just, this is the complete life history from the seed germination to senescence, right? That is called development, right? Senescence means death. So senescence means death. So the complete life history from the starting to end is called development, right? So please note it down. Development is sequence of event that occur in development is sequence of event that occur in life history of a cell, comma, organ development is the sequence of events that occur in life history of a cell, comma, organ or organism, which includes seed germination, comma, growth, differentiation, seed germination, growth, differentiation, comma, maturation, flowering, seed formation, and senescence. Senescence means death, and senescence. Within the development, we study inside the development, put a heading plasticity. Now, what is the plasticity? Plasticity means when a same plant is put in different environment, it have different type of the leaves, right? Sorry. So this is example of leaf actually. When organism is put in any environment, it adapt that environment and it changes its structure or physiology according to that environment. This property is called plasticity. So plasticity is nothing, the property which we call adaptation in animal, the same property in plant is called plasticity. So plasticity means a type of adaptation. Here is an example of plasticity is heterophily. Hetero means different and phily means leaves. So look, this term heterophily means different, right? Different, and phily means leaves. So different type of the leaf. So in heterophily, this is the coriander. So coriander, you can see there are 4 type of the leaf. This is the leaf of juvenile state. In juvenile state, they have this type of the leaf. In adult, they have this shade of the leaf. In terrestrial habitat, they have this type of the leaf. In aquatic habitat, they have this type of the leaf. So different type of the leaf on the basis of their age and on the basis of their environment. That is called heterophily. So please put the heading plasticity within the development. It's a part of development. So plasticity, the ability to change under the influence of internal or external stimuli. Ability to change under the influence of internal or external stimuli. Internal or external stimuli is called plasticity. Full stop. The example of plasticity is heterophily. The example of plasticity is heterophily. Full stop. Heterophily is the occurrence of different type of leaves. Occurrence of different type of leaves on the same plant habitually in different growth phase or under different environmental condition. Is it okay, heterophily? Did you got my point? Alvia and Mazia, keep on messaging me, please. Yes or no, in the form of yes or no. If you got the point, message me yes. If you didn't got, message me no. Please tell me, did you got what is the heterophily and what is actually development? Look, this is the one more part of development. What is the differentiation, dedifferentiation, and redifferentiation, right? And here you have to note down this one, right? So first I will explain you what is the uh, these all point, right? Differentiation, dedifferentiation. So what happened? It's also part of development. So a meristematic cell divide. So first it divide, right? So one cell divide into two cells. Now what happens? Cell grow. So growth takes place, right? So first division takes place, then growth takes place. After the growth, what happens? So this is the one cell. So after the growth, what happens? The cell will differentiate. Differentiation means cell will opt a particular type of the function, right? It will have a particular type of the function. It will become parenchymatous cell, it will become xylem cell, it will become phloem cell, whatever it want to do, it will become particular kind of cell, living cell, right? Not non-living cell. So it can become parenchymatous cell, right? It can become mesophyll cells, like green cell having chloroplast. So then what happened after during the differentiation, it opt a particular function and cell stop dividing. So division, they lose the capacity of division and they become a permanent cell, right? So that is called differentiation. In differentiation, they get a particular function, stop dividing, and it become permanent cell. After some time, what will happen? After some time, when they get the injury, so during the time of injury, they have to heal again. So how the healing will take place? Now these permanent cell, they will become, for example, they are parenchymatous cell. So what will happen? They will again become meristematic cell and start dividing. So this property is called differentiation. So from meristematic cell to becoming a permanent cell is differentiation, and from permanent cell to becoming again meristematic, reversible reaction. That's why we call it dedifferentiation, right? So example, during the when dedifferentiation takes place, during the repair time, when the cells regenerate and when secondary growth takes place, at that time dedifferentiation takes place. After dedifferentiation, when they will divide, again these dividing cell, right, they again get differentiated. So now we will call it redifferentiation. So from dedifferentiated cell, when they will divide, again these dedifferentiated cell, again they get particular function and stop dividing, they become permanent cell. So this is called redifferentiation. So this is the differentiation, dedifferentiation, and redifferentiation. Please draw as it is, then I'll explain, I'll dictate. Make it your notebook, then I'll explain you. Let me know when you note it down, Alvia. Message me when you note it down. Done? Azia? Noted? I hope you might have done that. Yes? Alvia and Mazia, please respond, please. If you note it down, please respond me. Start noting down, please. The process of first heading is differentiation. The process of division of labor is called differentiation. Full stop. During differentiation, cell opt a particular function. During differentiation, cell opt particular function. Full stop. They stop dividing. They stop dividing and become a permanent cell. For example, a meristematic cell. For example, a meristematic cell changes into parenchymatous cell. Changes into parenchymatous cell after differentiation. Now de-differentiation. When a permanent cell cells again become meristematic and it regain the property of division Full stop. The de-differentiated cell becomes meristematic cell. Full stop. Now redifferentiation. When de-differentiated cells divide and again opt a particular function, This is called redifferentiation. This is called redifferentiation, right? So this was dedifferentiation. It's very important. Plasticity is very important for your school examination. And this is division, differentiation, dedifferentiation. It is also very important, right? Now come to the part of phytohormones, means plant hormones. So plant hormones are also known as phytohormone. In case if you are not getting any points, please let me know. I'll repeat that, but you have to tell actually that if you didn't got any point, right? So plant hormones are also known as phytohormone, right? Plant hormone are also called phytohormone. They are five type of the hormones: auxin, gibberellin, cytokinin, abscisic acid, and ethylene, right? So start noting down. Plant growth regulator, PGR, plant growth regulator, or plant hormone, or phytohormone. There are five type of there are five type of plant hormones. Full stop. A plant hormone, now it's a definition. A plant hormone is a chemical substance other than nutrient produced naturally in plant which may be translocated to another region for regulating one or more physiological reaction when present in low concentration. There are five type of plant growth regulator. So please note it down. Auxin, second; gibberellin, third; cytokinin, fourth; abscisic acid, fifth; ethylene. Now start writing down one by one. So we'll study one by one. First auxin, then gibberellin, then cytokinin, and then so on. Right. So put first heading auxin. So start noting down auxin. Auxin is, so what is the function of auxin actually? Basically, the main function of auxin is that it increases the height of, like, length of cell, right? It increases the cell size, right? So basically responsible for cell elongation, right? And this is also responsible for movement of plant in the direction of light, like sunflower, right? So that is a basic function of auxin. So start noting now. I'll explain one by one every point. Auxin is weakly acidic. Full stop. Next point. It promote the cell elongation. Full stop. it should be present. It should be present at concentration less than 100 ppm, less than 100 ppm, less than 100 ppm for cell elongation. Next point. This is the first plant hormone. This is the first plant hormone to be discovered. This is the first plant hormone. That's it. Don't write anything. This is the first plant hormone. Right. Next, the chemical composition of auxin is, look, IAA and IBE. Both are natural auxin, right? So they are the natural auxin. They are the natural auxin. So basically, auxin found naturally in the form of indole acetic acid IAA and indole butyric acid IBA. So both of the question have been asked in medical examination. So please note it down. Now you have to note it down the lower one as well. Synthetic auxin. The synthetic auxin which we use in market and agriculture, that is a 2,4-D and 2,4-T. These questions have also been asked, right, in medical exam. 2,4-D is a synthetic auxin and the full form is 2,4-dichlorophenoxyestic acid, and 2,4,5-T is 2,4,5-trichlorophenoxyestic acid. Note down, please. Now start noting down the function. First function, please note down the function of auxin. First, it stimulates respiration. It stimulates respiration. Next, it enhances the metabolic rate. It enhances the metabolic rate. Next, it is responsible for cell enlargement. It is responsible for cell enlargement, which is the most fundamental it is responsible for cell enlargement, which is the most fundamental function of auxin. Next point. Auxin controls the xylem differentiation. Auxin controls the xylem differentiation. Auxin controls the xylem differentiation. Next. Now, what is the callus? So, next point is it is also used in differentiation of callus. It is also used in differentiation of callus. Now, you know what is the callus? Callus is actually, that is the mass of undifferentiated cell, right? And it is used in, actually it is made in tissue culture. In tissue culture, we take a mass of tissue and we use auxin and cytokinin, and with the help of auxin and cytokinin, we promote them to be divided, right? To get divided and get differentiated, right? So callus is mass of undifferentiated cell. It is made in tissue culture. Next point is it is responsible for apical dominance. Apical dominance means the plant will, the apex of the plant or apices of the plant, it will grow upward, right? Means the length will increase and it will suppress the horizontal growth. So horizontal growth will be less and vertical growth will be more. That is called apical dominance. So note it down. It is responsible for apical dominance. That is it increases the height of plant and suppress, it increases the height of plant and suppress the horizontal growth of plant. Next point. It is responsible for formation of female plant. Means if you spray oxygen, more and more ovary will be produced. It is responsible for formation of female plant. Next, it is also responsible for seedless fruit. That is also responsible for formation of seedless fruit. That is parthenocarpy, right? So it is responsible for formation of seedless fruit, and seedless fruit means parthenocarpy. Parthenocarpy. Now the next question have been put in the form of notes. Next point, please put in the form of notes because it have been asked in medical examination. Already asked question. It stimulate root formation in stem cutting. It stimulate root formation in stem cutting. So what is that? Suppose that you want to plant a rose, and so how does the roses are made in a garden? So in garden, the gardener, what gardener do? Gardener cut many pieces of a stem and then dip into the oxygen and then it implant the that stem cutting. So that's how it is. It stimulates the root formation in stem cutting part after dipping in oxygen, right? So this was all about the oxygen, right? Clear? Hope it must be clear to all of you. Next one is gibberellin. Put the heading gibberellin. Next hormone is gibberellin, right? First point. It is weakly acidic. So its nature is that this is the weakly acidic. It is weakly acidic. Full stop. It have ring shaped structure. It have ring shaped structure. Full stop. It is responsible for cell elongation and enlargement. It is responsible for cell elongation and enlargement. Let's talk. It increases the internodal length. Internodal length means, for example, this is a sugar cane. So, for example, this is sugar cane. So, this This is the internodal length, right? So this is the internodal length, right? Internodal, the part between the two nodes, right? So after the spraying gibberellin, the internodal length increases. So it can be used in the crops like sugarcane, bamboo. Internodal length will increase. So start noting now. It is responsible for internodal length. One more important thing which I want to share with you, that the gibberellin was discovered by a farmer. The name of the farmer was Kurushava, right? Kurushava, and he saw that in rice there is a disease called Bakane disease, and Bakane disease is also known as the English meaning. Bakane is a Japanese term, so English meaning means that is the fool seedling disease. So what happened during this disease? A fungus infect the rice plant, and a rice plant have a covering of the seed, but inside the covering there is no seed. So seed formation do not take place and productivity decreases. This is called fool seedling disease of Bakane or Bakane disease. So this disease causes by a fungus known as Gibberella fusacorae. From Gibberella fusacorae fungus, we have discovered gibberellin, right? So please note it down. The gibberellin was discovered by Kurushava. Gibberellin was discovered by Kurushava in Gibberella fusacorae. In Gibberella fusacorae, which is responsible for Bakane, or which is responsible for Bakane or fool's seedling disease. Now put the heading functions. Now what are the functions? So start noting down. In function, stem and leaf growth takes place due to it. Stem and leaf growth takes place. Next point. It increases the length of the stem. It increases the length of the stem. Next point. It is responsible for, third point, it is responsible for bolting. Now, what is bolting? Let me explain you what is the bolting. Look, this is the cabbage, right? Cabbage. So you must see that when we cut the cabbage for sake of making anything, so when we cut with the knife, it's cabbage look like this one, a small stem-like structure, and in stem-like structure, these are leaflet, right? So these are leaf actually, and these are small. This is a stem. This is a stem. So when we put the gibberellin in, so hardly the length is 12 inch or 1 feet, right? So when we uh, spray the gibberellin, what happened? It becomes like this way. Internodal length increases, so stem internodal length increases, and this cabbage become of 5 feet height, right? So the process of making genetically dwarf or roset-shaped plant in the plant which have greater height is known as, the process is known as bolting, right? So this is the actual picture of bolting. This is the, when we spray the gibberellin, the cabbage become like this one. This is a cabbage. Right? Cabbage become this like this one, 5 feet height. Right. So please note it down. It is responsible for bolting in. It is responsible for bolting in. Bolting in. It is responsible for bolting in genetically the dwarf plant. Next point. They help to come out of dormancy. They help to come out of dormancy of buds, tuber, or seed. Bud, tuber, or seed. Dormancy is that time period in which a seed or a bud keep on sleeping, it do not germinate, right? From that time, right? So, for example, what is the dormancy? This is the time to take out the potato from the field, and the new potato that you just take out from the field, and again sow it. It won't grow because now it will go under the dormancy. Suppose that we have harvested the wheat and you want to immediately after harvesting you want to grow the wheat, but you just sown the wheat, it won't germinate. So for a particular time period, for 5 6 months, and after 6 months if you will sow, they will germinate, right? So what they do in this 6 months, they keep on sleeping, and this duration of 6 months is called dormancy period, right? You can note it down. Dormancy is a time period in which seed do not germinate. Dormancy is a time period in which seed do not germinate. Next, it helps to seed germinate, especially in case of cereal. It helps seed to germinate, especially in case of cereals. Next, auxin and gibberellin, auxin and gibberellin are responsible for fruit growth and development. Fruit growth and development. Next point, put the heading as a star shape. This is the question for your medical. They promote the male flowers. Auxin promote female flower and gibberellin promote male flower. It promote the male flower formation. Hope it must be clear to both of you. Now next heading is cytokine. Put the heading cytokine. Cytokinin, it is basic in nature. Next point, it is responsible for cell division. Stop. It was discovered by Skoog and co-worker. It was discovered by Skoog and co-worker. Subheading, function. Function. So first function is it help in cell division. Next, responsible for cell elongation. Next, it help in differentiation of leaves and chloroplast differentiation. Next point, it also help in overcoming from seed dormancy. It also help in overcoming from seed dormancy. Next, it help in to provide resistant to plant, resistance to plant in low and high temperature. In low and high temperature. Next, it is also responsible for parthenocarpy. It is also responsible for parthenocarpy, that is seedless fruit formation. That is seedless fruit formation. Next is ethylene. So start writing now. Ethylene is a gaseous hormone. Ethylene is a gaseous hormone. Make a star. This question have also been asked. Next point. It stimulates transverse growth. So what is that? This is responsible for stimulating the transverse growth, and so it increases the size of branch. That's what I want to tell with you. So suppose that what happened? Suppose that this is the plant. So it is responsible for increasing the growth of branch. So help the horizontal growth, means it suppress the apical dominance and it do not allow the growth in a vertical manner, right? So note it down. It stimulate the transverse growth but retardate the longitudinal growth. It stimulate the transverse growth but retardate the longitudinal growth. Next, almost it is found in almost all part of plant. It is found in almost all part of plant, that is root, leaves, flower, fruit, seed, etc. Next point. It is responsible for ripening of fruit. Next point, it is responsible for ripening of fruit. So basically the fruit ripening is takes place due to them. Nowadays, that's how the fruit are ripened. So what they do, they banana and all that, they use a cylinder of ethylene, and they what they do, they put these fruits in an airtight chamber, and they open the cylinder of this is deodorant like small bottle, and they open the cylinder of ethylene, and the ethylene leaks out the whole night, and next morning all the fruits ripen. So that's how the fruit ripening is done nowadays, and this is not harmful. It's a natural. So function. They inhibit. So you have written down longitudinal growth. One more function noted down. They are responsible for. They are responsible for root initiation. Root initiation in low concentration. Root initiation in low concentration. Right. So one more, that is the abscisic acid. The next hormone is abscisic acid. Let me open the PPT. So start noting down. abscisic acid. It is also known as dormin because this is responsible for the dormancy. This hormone is actually responsible for the dormancy, right? And it is also known as stress hormone or drought resistant hormone. Why it is called? So that whenever a plant is facing a scarcity or drought situation, scarcity of water or drought situation, so what will happen? The plant will start releasing the abscisic acid in more and more amount. This abscisic acid closes down the stomata of plant, and by this way, it reduces the rate of transpiration. That's why it is known as state or drought resistant hormone. Please note it down. Abscisic acid, it is also called dormant. It is known as stress or drought resistant hormone. It's known as stress or drought resistant hormone. Full stop. Next one. It is also known stress or drought resistant hormone. Full stop. Writing on the same point. It is also known as stress or drought resistant hormone as during water scarcity. During water scarcity or water stress. During water scarcity or water stress, it is released in large amount. It is released in large amount and it closes down stomata and it closes down stomata to reduce transpiration. To reduce transpiration. Full stop. Next point. It was discovered by Edukiran et al. It's Edukiran and co-worker. It was discovered by Edukiran et al. Next point. It is responsible for bud and seed dormancy. Bud and seed dormancy. Next point. Next, you have written down, and next point. So, a small quantity of abscisic acid, a small quantity of abscisic acid causes flowering in short day plant, SDP. In short day plant like strawberry. Next point. It is responsible for rooting in stem cutting of roses. It is responsible for rooting in stem cutting of roses. This was the function of abscisic acid. Last but not the least, last part of that chapter, this chapter. So photoperiod. So photoperiod is what is that? Is the duration of light hours, right? Please note it down. Photoperiodism. 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 Please note it down. What is the photoperiodism? It is the effect of daily duration of light hours. Photoperiodism. It is the effect of daily duration of light hours on the growth and development of plant. especially in flowering. Next point. The phenomena was discovered by Garner and Allard. The phenomena was discovered by Garner and Allard in tobacco plant. The name of the variety of tobacco was Maryland mammoth. Right, Maryland mammoth was the tobacco variety of tobacco where photoperiodism was discovered by Garner and Allard. Right. So on the basis of photoperiod, there are three types of plants. Short day plant, they require a less critical duration of the light, less time period of the light. Basically, they flower in the time of winter time, this time. Next, long day plant, they require a light duration more than a critical time period, right? And like they flower in summers. And day neutral plant, they do not, they are not affected by the light duration. So put the heading short day plant, SDP. In short, they are called SDP, short day plant, SDP. They are winter flowering plant. Let's stop. They are winter flowering plant. Required short photo period for floral initiation. Short photo period for floral initiation. Example. Always remember the example. This is a question for medical. Xanthium, chrysanthemum, dahlia, rice, sugarcane, strawberry, potato, tobacco, and soybean. Next, long day plant. They are the summer. They are the summer and autumn flowering plant. They require long photo period for floral initiation. Let's stop. Example, henbane, wheat, oat, sugar beet, spinach, radish, barley, etc. Last one, day neutral plant. Please note it down. Day neutral. What is a day neutral? Please note it down. Third thing, day neutral. Day neutral plant. Please note it down. The plant flower throughout the year. Plant flower throughout the year. Example, tomato, pepper, cucumber, etc. Right? So There is one more thing, vernalization. What is the vernalization? So one more heading. Put a heading, vernalization. So put the heading vernalization. So vernalization is that uh, we reduce the flowering time period. So we reduce the, suppose that plant flower in 5 months. So if we when sowing the seed, so if we before sowing the seed, if we treat the seed at low temperature 0 to 5 degrees centigrade, their reproductive period means they'll flower in 2 or 3 months, right? So by this way we can reduce their flowering time period, so that this uh, low temperature treatment of the seed is called vernalization, right? Please note it down. Vernalization is a process of shortening the juvenile and vegetative phase, juvenile and vegetative phase, and hastening flowering by a previous by a previous cold treatment. Let's stop. The low temperature required for vernalization. The low temperature required for vernalization is 0 to minus 5 degrees centigrade. That's it. So this was all about this chapter, and this was all about the plant physiology and our syllabus, right? So today we have completed our syllabus, like complete syllabus of 11th, right? And for next class, like how the class will take place and when the class will take place, Nishkarsh ji will communicate will regarding the next class, like if we will continue with the revision, then what will be the mode of revision, what we are going to revise and all that. So regarding the class, you'll get the information from Nishkarsh, right? And in case if you have any problem right now, you can tell me that if you have any problem, any doubt, ask me. Alvia, Atif, Mazia. Any doubt? Any question or any query? Uh, you have asked for extra question, Mazia. Extra question, like chapter wise question. Okay, okay, fine, fine. I'll say to Niskarji. Definitely you'll get that. Definitely you'll get that. We'll arrange something, right? I'll discuss with Niskarji. And Mazia, please uh, uh, say Niskarji to give extra some questions, right? And in case if you have any problem, you can contact me as well, right? And he'll solve it, right? Fine. So any more doubt, any more questions? Okay. So see you guys in next class, right? Bye bye. Good night. Take care. Welcome, welcome. Bye bye.